Okay, today we are going to start with the past paper of chapter disease and infection, AS 9700. Now the question number one is the diagram shows the lungs tissue from a healthy person and lungs tissue from a person suffering from the emphysema. Emphysema means gross damage of lungs. तो ये वाली जो डायग्राम है ये नॉर्मल पर्सन की है और ये वाली जो डायग्राम है ये डैमेज जिस बंदे को जो है ना एम्फिसेमा हुआ है ग्रॉस डैमेज ऑफ लंग्स हुआ है उसकी है. अच्छा, describe two changes that can be seen between the lungs tissue of a healthy person and the lungs tissues of the person suffering from the emphysema. अच्छा देखिए healthy lungs tissue जो है उसमें number of alveoli जो है वो बहुत ज्यादा है, जबकि जो डैमेज लंग्स टिश्यूज हैं जो कि एम्फिसेमा की वजह से डैमेज हुए हुए हैं उसमें नंबर ऑफ alveoli डिक्रीज हो गए हैं. अपार्ट फ्रॉम दिस the spaces increased due to the damage of the alveoli in the emphysemic affected person affected lungs, और जो जैसे कहा जाए जो नॉर्मल है उसके अंदर जो स्पेसेस हैं alveoli की वो बहुत रिड्यूस है और छोटी है. अपार्ट फ्रॉम दिस blood capillary network is present in the normal lungs tissue, whereas there is an absence of blood capillary network in this emphysemic lungs tissue. So number one point is that number of alveoli decreases in lungs tissue with emphysema. And another thing is that thick walls of alveoli thick walls of alveoli develops emphysemic lungs tissue. एक इसका पॉइंट ये भी आप ऐड कर सकते हैं. इसके अलावा ये भी लिख सकते हैं कि नॉर्मल alveoli has proper blood capillary network around the external surface, whereas in emphysemic lungs tissue alveoli lack of blood capillary network. Okay, now another question related to this topic is explain how changes in the lungs tissues might affect breathing and gas exchange as the alveoli damages. So the person faces shortness of breath and oxygen depletion in body results in high breathing rate and less oxygen volume is transported to blood, as well as less removal of carbon dioxide or bicarbonate ions takes place. Okay. Now the next question is explain why the person Now the next question is, explain why the person suffering from emphysema may find it difficult to walk quickly. As we know that due to emphysema, gross damage of alveoli reduces the transport of oxygen in the blood, which directly affects the aerobic respiration and ATP synthesis. And this is a major cause that the person won't be able to walk quickly, and which results in lack of energy. अच्छा, as far as the lines and the space is concerned, actually ये जो है एक जिसे कहते हैं कि हमारा सॉफ्ट कॉपी व्यू है और डिजिटल व्यू है, जिसके अंदर पेपर की जो लेंथ और ब्रेथ है इट्स चेंज्ड. तो जब फिजिकली हमारे पास पेपर आएगा और उसके जो है ना जो लाइंस और उसके ऊपर जब हम लिखना शुरू करेंगे तो दैट मच मटेरियल इज इनफ टू फिल द टू लाइंस. And all the things are easily adjusted, all the concepts are easily adjusted in between these two lines. Okay, another question is, give 2 ways in which the risk of developing the emphysema may be increased. Number 1 is the smoking, and second one is the air pollution. Air pollution means dust particles or aerosols. Now another question related to the heart disease. The diagram shows the healthy artery and an artery from the person suffering from arteriosclerosis arthroma. ठीक है, अब ये वाली जो है ये देखो ये हेल्दी आर्टरी है. ठीक है, अब इसमें जो है ल्यूमेन जो है वो ब्रॉड है, पूरा थिक, जिसे कहा जाए कि फाइब्रस कोट है. एवरीथिंग इज स्मूथ एंड इलास्टिक. ठीक है, जो एक नॉर्मल आर्टरी के अंदर होता है, बट दिस वन इज फिल्ड विद द फैटी डिपॉजिट्स. ठीक है, फैटी डिपॉजिट्स. अच्छा, ये जो है यहाँ पे इसकी वजह से इसका क्या है, ल्यूमेन जो है वो डिक्रीज हो गया. इसका ल्यूमेन सिर्फ इतना सा रह गया. ओके, और ये वाला जो है ये ल्यूमेन काफी बड़ा है. डिस्क्राइब हाउ आर्थरोमा हैज अफेक्टेड स्ट्रक्चर ऑफ आर्टरी वॉल. आर्टरी वॉल become stiff, and another point is that reduced lumen can rupture the artery if that is passed with pressure. Okay, now another question is describe and explain the effect of arthroma on blood pressure. As lumen decreases, the backward blood pressure gradually develops and cause angina attack. Suggest how smoking may increase the risk of arthroma. Smoking contains different carcinogens carcinogens, including lead, which increases the accumulation of cholesterol and fatty deposits in the endothelium layer. Explain why changing from eating animal fats to eating plant fats decreases the risk of developing arthroma. As animal fats are highly saturated and difficult to digest, whereas plant fats are unsaturated, so easily digested and and do not deposit it. Okay. The coronary artery supplies blood to a tissue of the heart. Explain why the formation of arthroma in coronary artery may lead to the heart failure. As we know that coronary arteries are major supplier of oxygen, glucose, and amino acids for the heart muscles. If there is a blockage, then that reduce supply of O2 and glucose may lead to myocardial infarction or angina pain. Okay, another question is, some people may be given a heart transplant to replace the damaged heart. Explain why a transplant heart may be rejected. As a transplanted heart even from sibling or parental donor does not matches 100% acceptors genetic makeup, which leads body to recognize heart as foreign body and reject it by producing antibiotics. Okay, now question number 4. Tuberculosis is caused by a bacterium which lives in the lungs and cause the destruction of the tissue. Tuberculosis bacteria exposed to the air can form resistant spore. Overcrowded living conditions encourages the spread of tuberculosis. Okay. Describe how tuberculosis may spread from person to person by sneezing, coughing, air droplets containing infected bacterial spores. बैक्टीरियल स्पोर्स, क्योंकि ये बैक्टीरिया जो होता है जब इसको सुटेबल एनवायरनमेंट नहीं मिलती है, जैसे ड्राई एनवायरनमेंट है, तो उसमें ये स्पोर्स की फॉर्म में आ जाता है. एक्सप्लेन व्हाई ट्यूबरक्लोसिस इज स्प्रेड मोर इजीली इन ओवरक्राउडिंग लिविंग कंडीशन. क्लोज कॉन्टैक्ट as well as small area sharing by population. Okay, another question is during 1950 and 60. During 1950s and 60s, when tuberculosis was very common in United Kingdom, buses and railway carriages often had a notice saying no spitting. Explain why. Because sputum of infected person contains high number of TB bacteria which was the main cause of spreading infection. The destruction of lungs tissues by tuberculosis often leaves scars. Suggest why. अच्छा, इसमें स्कार नजर आते हैं बिकॉज वहाँ पे एल्विलाई जो होते हैं वो खत्म हो चुके होते हैं और हार्ट टिश्यू स्ट्रक्चर जो होता है वो फॉर्म हो जाता है. X-ray are often used to detect tuberculosis because X-rays efficiently absorbs at damaged tissue of lungs and produce dark circles where infection was prevailing. Mass screening by X-ray was successful in reducing the incidence of tuberculosis during 1950 in the United Kingdom because this method was quick in detection and less time taking and cost effective, very cost effective. Vaccination is used to control the spreading of tuberculosis. Give 2 reasons why children should have been vaccinated, even though the tuberculosis is now not rare in United Kingdom. To prevent spread of infection through air aerosols, अगर वहां पे है हवा पे कहीं इधर से आ जाए तो, और if a person travels to area where TB rate is high, so vaccination gives protection. Another thing is people who develop tuberculosis are treated with antibiotics sprayed by as TB bacteria are living organism, so antibiotics can kill them and eradicates them. Okay, the diagram shows the structure of HIV magnified 10,000 times. Glycoprotein spikes, outer coat and inner coat, nucleic acid and 100 nanometer magnification इसके लिए that means इसको magnify किया. Okay, HIV is a retrovirus. Name the nucleic acid present in HIV RNA. One of these genes of HIV codes for the production of reverse transcriptase. What is the role of reverse transcriptase? It converts virus RNA into virus DNA for replication. Name the cell which are infected by HIV. Helper cell or T cells और T lymphocytes. ये सारे एक ही नाम. Okay, describe how HIV replicates in these cells. Describe how HIV replicates in these cells. The RNA of virus when enters in the T lymphocyte due to reverse transcriptase it converted itself into viral DNA which starts replicating HIV new viral particles in the T helper cells and comes out in the form of budding, taking the outer coat of glycoprotein from the cell membrane of T lymphocytes. That is why immune system जो होता है वो इतना ज़्यादा effective नहीं होता इसमें क्योंकि जो इसकी outer covering होती है उसमें glycoprotein more or less similar उसी structure का होता है जिस structure का lymphocytes पे composition होती है glycoprotein की. तो बॉडी जो होती है वो उसको फॉरेन पार्टिकल के तौर पे रिकॉग्नाइज नहीं कर पाती कि हेल्पर सेल्स की वैसे ही तादाद कम हो रही होती है, तो एंटीबॉडी प्रोडक्शन ना होने के बराबर होती है, और इसको रिकॉग्नाइज जो है वो इतना इजीली बॉडी नहीं कर पाती। एंड वो जो है ना फिर यही होता है कि वो एक सेल से दूसरे सेल, दूसरे से तीसरे, तीसरे से चौथे मतलब लिम्फोसाइट्स को इन्फेक्ट करता रहता है और उनको तोड़ तोड़ के निकलता रहता है, और स्टेप उसका ये होता है कि आपका ग्लाइकोप्रोटीन जो होता है वो उसको बचाता रहता है। Now explain why an HIV infected person may not show any effect for several years. HIV का जो वायरस होता है उसकी सबसे बड़ी क्वालिटी होती है कि sometimes it gets entered into the body by some or different reasons जो कि डिफाइन किए गए हैं, तो बॉडी के अंदर जाने के बाद वो मल्टीप्लाई नहीं करता है, लैटिन जिसे कहा जाए कि एक तरह से लैटिन पार्टिकल की तरह जो होता है बिल्कुल डॉर्मेंट फेज के अंदर वो पड़ा रहता है, ठीक है। वायरस do not show any replication, latent for several years. Okay. As I explained in different ways in which the spread of HIV can be prevented. Number one, remain to one sexual partner seeing blood uses blood mate. Use for blood transfusion. Okay, today we are going to start with the another past papers of disease and immunity. The question is, the diagram shows the cell of the bacterium. Yes, the diagram shows the cell of the bacterium. The site of the action of two antibiotics is labeled on the diagram. One is chloramphenicol, which inhibits the protein synthesis and ribosomes, and another one is the penicillin, which inhibits the synthesis of bacterial cell wall. Explain why penicillin is safe to use in the human body to treat the disease caused by the bacteria, because of the fact that penicillin only acts on the cell wall material of the bacteria, and it's never ever harm to the gives never ever harm to the cell membrane or any mammalian cell organelles or mammalies or any part of the cell of the mammals. So it is safe to use. That is why it is safe to use. Penicillin. Penicillin only attacks or inhibits cell wall synthesis of bacteria. Chloramphenicol may have side effects affecting the bone marrow. Why? Because of the fact that chloramphenicol stops the or affects the protein synthesis of the cell. That is why it also affects the protein synthesis of the stem cells of the bone marrow, and that is why it affects on the normal bone marrow cells of the human body too. As chloramphenicol may stops or hinders the protein synthesis of bone marrow cells, which causes anemia and leukemia in a person during its use. Another antibiotic, actinomycin D, prevents the replication of DNA. This antibiotic is considered to be too toxic to human to use against bacterial infection, but may be used in chemotherapy to treat the cancer. Suggests why actinomycin G be an effective anti-cancer drug, because it stops the replication of DNA in fast growing cells. So it can also use as anti-cancer drug, because it stops cancerous cell DNA synthesis. DNA. Explain how monoclonal antibodies can be used to target anti-cancer drugs more precisely at cancer cells. As monoclonal antibodies are capable to attach with the surface area of cancerous cells, and also capable to attach with almost all type of cancerous cells, so they can be used as a good source for treating cancer. Next question is, give one reason why antibiotics cannot be used to treat viral disease. Viruses are not included in living creatures and do not show any living metabolic process. So antibiotics are useless act on them or destroy them. The virulence of pathogenic microorganism depends upon two properties, their invasiveness and their toxigenicity. Bacteria may produce either exotoxin or endotoxin. Define the term virulence. Capability of any microbe to cause infection or disorder in the living body. Distinguish between invasiveness and toxicity. Invasiveness means ability to invade in the host cells or tissues to cause infection. Okay, and toxicity ability to produce toxin in order to harm the host cells and tissues. Distinguish between exotoxin and endotoxin. As the name shows, exotoxin means the toxin that has been secreted out or excreted outside the cell of the pathogen, and endotoxin is means that is present inside the pathogen cell or body, or any part of the pathogen may act as a toxin and can cause the disease or the infection uh, in the host body. Exotoxin toxin that is secreted out by pathogen to damage host cells or tissue. Endotoxin, any part of pathogen that can cause infection or responsible to cause infection. Now, exotoxin and endotoxin example, exotoxin like Staphylococcus toxin, exotoxin या फिर कॉलरिनोजिन कॉलरा में जो प्रोड्यूस करता है बैक्टीरिया, और एंडोटॉक्सिन में आप कह सकते हैं साल्मोनेला दैट कॉज टाइफाइड. Capillaries are known as exchange vessels. Substances are exchanged between blood and tissue fluid as the blood flows through the blood capillaries. Figure 1.1 is an electron micrograph of a section through a capillary with two red blood cells. ठीक है, blood capillary है और इसके अंदर दो red blood cells है, एक ये हो गया और एक ये हो गया. अच्छा, अब इसमें चलो कुछ लेबलिंग है. It's a part of A and part B. Whereas the part A is the endothelial or squamous lining or the tissue of the blood capillary, and B is the nucleus. It's a endothelial smooth muscle lining and B is the nucleus. Another one is about the magnification and the measurement and ये वाला जो सवाल है ये उसी वक्त प्रॉपर्ली सॉल्व हो सकता है जबकि हार्ड कॉपी जो है वो आपके पास मौजूद हो. सो पार्ट 2 जो है वो हम ऑनलाइन जो है उसको सॉल्व नहीं कर सकते हैं. Another question is explain how capillaries are adapted to their functions as exchange vessels. As we know that blood capillaries are very thin single cell layer vessels and it is rich in intracellular spaces which supports the passage as well as exchange of gases by diffusion and nutrients and waste by process of simple diffusion and osmosis. Table 1.1 shows the composition of blood tissue fluid and lymph. Components that is red blood cells, white blood cells, glucose and protein, and their ratios or amount is given in the blood as well as in the tissue fluid and the lymph. Now they are asking about explain the difference between the composition of blood, tissue fluid and lymph as shown in figure 1.1 for white blood cells, glucose and protein. Now first of all we have to observe the chart and see the chart and write it what is high, which factor is high and which factor is low in blood and tissue fluid and lymph. First of all the WBCs that is white blood cells. So अगर हम WBCs और white blood cells को study करें so the highest ratio present in the lymph because they are synthesizing in the lymph nodes or lymphatic vessels and run in the inside the lymphatic vessels. So highest number present in lymph and second highest in blood because blood is a transporting medium तो ये एक जगह से दूसरी जगह इनको ट्रांसपोर्ट भी करते हैं as far as lymphatic vessels are concerned वो भी ट्रांसपोर्ट कर रही होती है लेकिन blood जो है phagocytes जो बोन मैरो से सिंथेसाइज होकर आ रहे होते हैं वो भी जिसे खाते ट्रैवल कर रहे होते हैं तो सेकंड हाईएस्ट जो नंबर होता है इनका वो ब्लड के अंदर होता है एंड थर्ड वन इज द जो सबसे लोवेस्ट रेशियो होता है या लोवेस्ट नंबर जो होता है वो टिश्यू फ्लूइड के अंदर होता है एंड लोवेस्ट इन टिश्यू फ्लूइड. अब इधर इतना कम इसलिए होता है बिकॉज जहाँ जहाँ इन्फेक्शन होगा वहीं वहीं ये टिश्यू फ्लूइड के अंदर प्रिवेल करते हैं अदरवाइज दे नेवर एवर get inside the tissue fluid. Another one is the glucose. So the ratio or the amount is same in blood as well as in tissue fluid. Blood and tissue fluid has same amount of glucose and in lymph it is slightly low as compared to the amount of glucose in blood and tissue protein. थोड़ी सी कम है उसमें. Outline how red blood cells are involved in the transport of carbon dioxide. When carbon dioxide enters in RBC, which will form carbonic acid, पहले तो वो carbonic acid बनाती है water के साथ मिलके when it dissolves with the water in the presence of enzyme that is carbonic anhydrase. ठीक है, enzyme कौन सा रहता है? Carbonic anhydrase होता है और ये एक reversible reaction होता है. This carbonic acid is disassociated into hydrogen ions as well as bicarbonate ions and removed from the RBCs by the help of chloride shift. ठीक है, chloride shift होता है जिसके अंदर सोडियम बाइकार्बोनेट और पोटेशियम बाइकार्बोनेट फॉर्म होता है. क्लोरीन जो कंसंट्रेशन अंदर ज्यादा हो जाती है और इस तरीके से कार्बन डाइऑक्साइड जो होती है वो रिमूव होती है अल्टीमेटली both in the form of salts as well as in the form of a dissolved form of a gas. On the other hand, carbon dioxide in RBCs combined with hemoglobin to form carbamide hemoglobin, which is the temporary component and can be disassociated easily. फिर वो easily जो होता है वो disassociate होता रहता है और ये भी एक reversible reaction होता है. That's how carbon dioxide is removed from the RBCs and from the blood. Okay. Figure 4.1 shows the two base pairs in DNA in a DNA molecule. This one is a pyrimidine and this one is purine. One is labeled that is adenine, double bond है, that means this one is the thymine. And this one is a triple bond है, तो big molecular structure जो है वो है K, guanine, and this one is the cytosine. सबके नाम पूछे गए नीचे, वो मैंने यहाँ पे लिख दिए हैं. Okay, HIV enters T lymphocytes by a form of endocytosis. Two of the enzymes in HIV are reverse transcriptase, which uses viral RNA as a template to make the DNA to incorporate into the chromosome of the host cell. ठीक है, वो अपने आप को replicate करवा देता है और RNA से DNA बना देता है. Protease, which is used to break the polypeptide into smaller molecules. These molecules are used to make the protein coat of new viral particles, which will infect the other cells. Various drugs have been developed to treat HIV infections. Table 4.1 gives the information about some of these drugs. Drugs azithromycin reverse transcriptase को inhibit करती है. That means कि RNA को जो है ना DNA में convert करने वाला जो enzyme है उसको inhibit कर देती है या खत्म कर देती है. Occupies active site. अच्छा, इसका mode of action जो है वो active site पे फिर जाके attach हो जाती है, जहाँ से ये reverse transcriptase synthesize हो सकता है. अच्छा, tenofovir reverse transcriptase same occupies the active site. फिर Efavirenz reverse transcriptase occupies sites other than the active site. ठीक है, दूसरी sites के ऊपर जाके attach करती है other than active site. Ataxanthin protease, इसके protease की synthesis जो है उसको inhibit करती है और enzyme synthesis जो होती है और उसके बाद it occupies the exactly active sites. Explain the difference between the mode of action of zithromycin and efavirenz. ठीक है, zithromycin जो है उसका mode of action ये है कि it occupies active sites and efavirenz reverse transcriptase को ही रोकती है लेकिन it occupies other than the active sites. जितना इसको पूछा गया है बस इतना ही बताना है, इससे आगे हमें explanation नहीं देनी है. That's why हम सिर्फ इतना ही बताएंगे. People who receive drug treatment of HIV take the mixture of the drugs that acts in a different way. Suggests the advantage of taking the drugs in the form of a mixture shows in figure in table 4.1. Actually, इसके बहुत सारे reason होते हैं. First biggest reason is that person may get different strain, multiple strains, may get multiple HIV strains strains in your body, or HIV or the virus may mutate or change its um, shape and outer covering coat for a specific drug. तो दूसरी जो कोई दूसरी ड्रग जो होगी जो मल्टीपल दी जा रही है उसमें से दूसरी वाली जो है वो अवेलेबल हो जाए। तो ये दो रीजन जो है